going here? Okay. Good evening, everyone. This is Taxonomy Tuesday from New Jersey Mycological Association. This is where our members share their recent fungal finds. If you have something to share that you haven't sent in an email to Taxonomy Tuesday, please put your name in the chat and we'll get to you in order of who's available and online and ready to go. Would anybody like to start? Is she on yet? Is she hung
I
on
got a
yet?
couple.
Okay, Chris, why don't you start? You had the largest fungi found this weekend.
Yeah. So other than getting rained on this weekend, we also took a much drier walk at Clayton Park, and there was lots of good stuff around. The
Is
one,
that also in Morris County or is this someplace else?
excuse me?
Where is Cream Ridge? Is that Morris County also?
Oh, that's Monmouth.
Monmouth, okay.
I
Clayton
think.
is a mammoth?
No, maybe that's the very top of Ocean County.
I thought it was Burlington, actually.
No, it's not that far over.
You're talking about the place below I-195, right?
Yeah, it's right there near not far from
Exit
Six
11.
Flags. Yeah,
Yeah,
exit
exit
11.
11, yeah. Ismail Town.
Right. How did you just say that?
Ismail Town?
I
That's
don't
that's
know.
my
Maybe you're
that
right. Every time I see that sign, my brain doesn't want to do it.
yeah, it's my version of pronunciation, but
As good as any, I suppose. But I saw these, and after being at the 4A, my brain was, I swear, not going, and all I could get out was G rufa, and I was just happy I remembered.
Yay!
Your picture's much better, Hunter.
But you found it too. That's nice.
There was a couple of these.
It was just a nicer specimen, not a better picture.
is that a Pleurotus levis?
I believe so. And boy, look at all of those beetles. It
So
was positively crawling.
somebody's having too much fun.
But I could swear I found these things on the same tree last year.
I
It
think I
looked,
may have seen those. What's that? A cedar?
no, I think it's a maple.
Maple, yeah.
Huh?
Yeah, that's what we've been finding these on here in Northeast PA, on usually on maple. It seems to be a good year for this species.
This is the only park I really find it in, close to me. Uh, oh, some some stidii. Eh, Uh,
Well, yeah, I saw my first one too this last week.
some pretty good chunks of it, all bruised up.
I mean, the browning when you touch it, right? Black
Yeah,
staining
black
polypore.
staining.
Yep.
Really.
Sometimes you have to wait for a while for the stain.
Yeah, that one actually did take a while. I took that chunk and I just kind of kept walking with it for a good, 5 minutes before it really.
Yeah, sometimes longer than that. If you rub it, it helps.
think this is the cinnabarina, Trametes.
So is that on buried wood there?
No,
Kind
it's
of looks
on
like the
that
other one.
branch.
Little.
yeah, it's a cherry branch of some sort. Might be a black cherry. I mean, the only other thing that's in those woods besides a couple of those are birch trees, which I'm not sure if that thing grows on birch trees, but the stick, I should have got a better picture of it. You could see all the little lenticels in the stick.
Yeah, I would say the most common substrate I find this on is black cherry. I've also found it on beech.
Well, there's plenty of those around, but they don't have lenticels,
What
right?
Oh,
lenticel?
right. Yeah. Yeah, sure. Yeah,
What is,
it's probably black cherry.
Yeah.
what is a lenticel?
I in this picture you really can't see. If you look at any cherry tree, the little horizontal lines, they're like almost like little tears in the bark.
Oh, okay. Yeah, I know what you're talking about.
And birch trees have them in the same way. They go, horizontally.
Okay. Do you get buckthorn there?
I don't think so.
Oh, good.
We have it in New York State, big time.
Oh, does it have lentinos? Or
Yeah, I think the younger bark might.
I think so too.
Yeah, that has a shiny bark and the shiny leaves. That's how I know that.
Okay. I do. I know it too, unfortunately. What's this, Chris?
I'm not sure. These were you could see they're all only about, I don't know. I think that biggest one in the middle is maybe 2.5 inches tall tops. Uh, the two on the right there have some kind of hypomyces, which there was a ton of these littered on this one section of trail, all about the same size,
Can we please
and
zoom in?
half of them were completely eaten up with the hypomyces.
Is this some kind of a Gyroporus?
It has pores, right,
I
Chris?
think this. Yeah, I think
Oh,
this is
yeah.
Gyroporus. Yeah.
Yeah, so one thing you could check if you think it's Gyroporus, you can see if the stem is chambered.
Yeah, you can see those scales on the stipe, like it's like finely rhymose. That's
Yeah.
what
Yeah.
over Gyroporus. Yeah, this is Gyroporus. Whether it's Smithii or
Yeah, it
something
looks
else.
like it looks like the cap broke off the one also.
Yeah,
yeah.
that's actually what drew my eye is the cap was upside down. So it was like it was like a bright white spot in the leaves.
Yeah, Gyroporus has brittle flesh. It breaks pretty easily. No staining.
I didn't cut one open, but I figured if the top was broken off of that one, maybe there would have been something
Yeah.
there. And
No, these don't stain.
Right. Yeah, these don't. Right.
Wow.
So this is chestnut bolete, which is several different species in eastern North America.
They didn't
But
get
but it's
much.
not castaneous, interestingly.
Yeah, that's what they say. I guess they haven't found that at all in North America.
No, no,
No. Oh, okay.
no, not to my knowledge.
So it might be Smithii. That's one of the names. There's a couple of newer names. Borealis is a name, but I think that might be the dark one.
yeah,
Yeah,
I think that might.
I'm still waiting for somebody to sequence ITS and TEF such that we can compare apples to apples, because the paper that published these names by Navid
Yep.
Davudian,
Yeah.
utilized other loci, not ITS. So we still don't have any knowledge as to what Smithii is in terms of ITS and LSU. So
So
all of
you
that
said
information is absent.
that stipe would have been chambered. I mean,
Yeah, it's
like
kind of hollowed out in a very kind of like bizarre way, kind of cavernous.
Yeah, caverns. And when they're young, the caverns are kind of stuffed with a pithy substance. So they're not like completely hollow.
all right. I'll have to, if I see those again, I'll have to chop one
Yeah,
open.
see if you could snap the stalk in half like a piece of chalk, and then you'll see one of the chambers. So most boletes don't have stalks that snap. They're a little more fibrous. And Gyroporus species have brittle flesh, so the stipes snap apart.
Yeah,
And
it's
they're
a good
good
it's a good
edibles.
edible, but you should discard the stipes and just eat the caps.
Huh. All right. These, as you can see, are in my wood chips again this year. These Chlorophyllum, I can't ever pronounce that.
Molybdites or molybdites.
Molybdites, yeah. But since they're at my house, I got a good bunch of. So the last one was in the morning. This one's in the afternoon, and then the next day, in the morning, I had to stop my car as I was leaving for work, and got this shot. And then when I got home from work, wait, no, that one's still in the morning. That's in the afternoon. They're starting to open up a little bit more. And they have this again this year. This, marginate gill.
Yeah.
So
I don't know. Maybe it's just because they're sitting there getting beat down by the sun, or
Mm-mm.
you get a spore print for these, or maybe last year you did.
Yeah,
oh, they're the greenish.
Okay.
Yeah.
Huh, maybe that's a species complex, because I don't think that's a trait that's reported typically for molybdites. Marginate gills, that is. I would have to look. I don't find that up here. I only found that once in Luzerne
We
County.
we talked about it last year, and everyone seemed to think that wasn't a thing, and that they've never seen it before. So it's just interesting that it's it's definitely there again.
Yeah, with that same trait.
Yeah,
That's
the exact same thing.
pretty interesting, really.
But that's all I had.
Man, those have been absolutely everywhere where
Oh
I'm
yeah.
living in South Jersey. I'm in and out of neighborhoods all day long, and I see them, 5 times an hour when I'm driving around. So
they're so dramatic. It's so fun to see them when they're often in fairy rings, and when their caps are fully open, they're really big.
oh yeah,
It's
you
the
can't miss. They're not they are conspicuous.
I don't see a lot of people kick those ones over, honestly. I think that, like, you'll see them on the lawns for a couple days if you travel the same place or whatever. People don't seem, but other mushrooms, people seem that they can't wait to just, I got to get rid of that thing.
No, I've seen a couple piles of them at the curb.
Oh,
Someone was, I don't know what these are. We got to get these 30 chlorophyllums off the lawn, and they just put them like where they would put their leaves.
If you have a pet or a child that frequents the lawn, it's probably a good idea to do that.
Yeah, that is true.
Well, with a name like Vomiter.
Yeah, you definitely don't want to eat those.
They sure are pretty, though.
Yeah, I love to look at them. I just checked a couple of my observations, and yeah, I don't see marginate gills on mine. I think you should save one and get it sequenced.
Yeah, I'll dehydrate one.
Okay, thank you, Chris. Does anybody else would like to go that hasn't sent in an email?
I can go eventually, but I can wait. I'd rather wait.
Okay. Brandon, would you like to go now, or you want to wait also?
No, I can go.
Okay. Still with you then.
Yeah, so I sent in. I went out Sunday and I found just so many slime mold. I could have set in 10 observations of different species of slime molds. This is one. This is in the genus Cribraria. And if you can click on the photo, it'll enlarge and you can kind of make out some details.
I'm in charge here. All right. Moving that. Sorry.
Very cool.
Moving.
Is that blown out?
Yeah, that was what I was going to show on the first photo. So the first photo is with the spores in the sporocarp,
Okay.
and they're
Cool.
yellow. It's the foot. It's got a little bit of I don't know if that's lime deposit on the foot or not. That's kind of one of the ways to work out different genera of myxomycetes, but I don't think it is. I don't think that's a Cribraria thing.
Lime
Is that
deposits.
Yeah, lime.
Neat.
Yeah.
is that similar to like the little crystals that you find in some of them?
Mm-hmm. Yep. Yeah. Some. I think it's actually only reserved to Physarales, but I could be wrong on that. But the way to determine between species and genera in Physarales is whether there's lime in the peridium and the foot, one or the other. So,
Cool.
but Cribraria, I think I shared an observation of a calcareous one. But the thing to note on the macro are the nodes, and when I say macro, I meant the other photos. This is what it looks like in situ, so not much information. But under the stereoscope and the microscope, you can see these nodes.
Ooh,
Yeah,
look at
and if we
that.
can zoom in a little bit, which just click on the photo. So there's all these interconnections, right? And the node. I think we all know what a node is, right? Where all those interconnections meet, the size and the frequency of the nodes, and how many kind of limbs it has coming off of it. That's all important information when you're looking at some of these slime molds.
It's like the Epcot Center of slime molds.
It's fair. I like that.
Need to load tonight. Yeah, okay, this is a close-up I missed.
Yeah, just trying to catch how many different, arms this one node is sending out in this photo. And you can catch a spore that is relatively in focus and a decent measurement. You're not getting much of the ornamentation on these because I don't look at the spores at 1,000. I just look at them at 400, and they're all subglobose. They pretty much all have some kind of ornamentation. But yeah, Cribraria. It's a cool genus.
What was that growing on?
Decaying hardwood.
Oh, thank you. Can you blow that up too, just for a sec?
They are small. I didn't include a quarter in this photograph, but this is one of the larger Cribraria, and I don't know the species. I've been trying to figure it out, or similar observations and collections. There's a couple options that none of them really fit the bill, but
Very nice.
all right. And then this is a Tubifera. And I won't spend too long going into it, but Ferruginium, Tubifera ferruginium is a species complex, and there's more out there. I'd love to just call this Ferruginium and move on, but I don't think it's the true Ferruginium sensu stricto. And if you can click on the photo and then just cycle through using the arrow so you don't have to go back every time.
Yeah, yeah, I'm just trying to get it to enlarge. My internet is being very slow today. I'm still waiting for it to enlarge here. There we go.
Oh, wow.
So I
It's
guess,
got the faded.
I was just, is this like some, relative of the chocolate tube slime or whatever?
I have to look that. I have to look that up. I would say no. Whether or not this is in Stematales, I mean, I'll look it up real quick.
'Cause these
And
don't
that
have
could,
stalks at all.
well, if we can cycle through the photos using that arrow.
Oh, wow. Oh my
So
gosh.
these are like mid maturity. And you can see they're individual sporocarps that are just clumped together, right? This isn't branched like you maybe were alluding to there, Chris. Like it's not a bunch of things coming off of one branch. So that's a staminatales thing. But if you go to the plasmodiocarps, the, the yeah, just the next photo. Oh, look at the size of these, which for Tubifera, they're really small clumps, which is pointing me in a direction of something within Ferruginium complex that might be an interesting name. But yeah, I'm not exactly sure what these are, but they're really small clumps.
Yeah, Ferruginium is usually like pink or red, right? At least at one stage.
Yeah, and the plasmodial stage, right? But as they mature, even Ferruginium sensu stricto becomes brown.
Oh, okay.
Oh yeah, yeah. So this is that plasmodial stage, and it's pale even for Ferruginium, which is why I was thinking Tubifera magna, which is a different species within that complex. But yeah, the true ferruginium is normally super bright red, like Corvette fire truck red.
And they have that same white foot to the bright red ones.
Yeah, but it's less pronounced, right? Because those plasmodiocarps are they're larger. Um, so it's spread out amongst a larger surface area. So in this one, it seems more pronounced, but they have that white foot for sure. Pseudothallus, I
So
think.
so those red ones, I've never seen them in that end stage, I guess. You're
Yeah,
saying they're red and then they change.
yeah. I bet you if you ever see that red stage, you could, if you know what you're looking for, you could probably find some mature brown fruiting bodies.
I gotta go slower.
Uh, but then the newest paper that I've read on this uh, complex is it's all about the size, the fruiting body, and the shape of the fruiting body, and then the apex of the mature individual plasmodia. So I don't know if you can get to that photo. So
Should I go left or right? It
right,
takes so long to load. Right.
yeah, it's probably, I think it's the second to the last photo, but yeah, there you go. So you can see these are kind of rounded, somewhat truncate, and they have a kind of unique way that they're opening up at the end. But yeah, we can move on.
Did you take that
Waiting for it to
home?
load.
I'm
Did
sorry.
you did you take it home?
Yeah,
Yeah. Did you take the substrate with it then?
I did not. No,
You did not? Oh, okay.
no. I've never had one of these fruit in any of my moist chamber cultures, but I have similar substrate. So
Oh, okay. So you're gonna keep it as a pet?
yeah,
Good. That's exciting.
Ready to go on to the next one, Brandon?
yeah, that's what I meant when I said we can move on.
Okay.
What?
Oh, cool. Wow. Amazing.
Yeah, and just now realizing that we were talking about the Cribraria, and when I was talking about the lime, I was unsure whether or not I shared this, but this is something in Physarales on a sweet gum.
That's very neat.
Yeah, I thought this was I've never seen this before, so I thought it was really cool.
Really.
That's great.
Well, photo contest.
Yeah, I like the first shot. It's like a disco ball, but a mixco ball.
Oh, that's the name
Yeah,
on the photo.
lame. All right, but zoom in on this photo, and you can see that they're little, you know, uh, little sporocarps with a little foot. They're pretty small. And it's not just on the sweet gum seed pod. It's on the leaf in the background. It's grown on the stem of the seed pod. So it's not like host specific on, sweet gum seed pods. It's just it liked this area and whatever it was feeding off of liked this area also. But it's not necessarily feeding off of the sweet gum seed pod or leaves or anything like that.
Neat.
And reddish. My this scope doesn't do a great job of color, but I think it's from what I could see in the field and through my loop and things like that. The stalk was red, and the hymenium, the fertile ball on the top of the stalk, uh, was tomatos squamulos, which maybe you can see better in the next photo. Yeah, and a lot of those kind of protruding bits, I'm pretty sure are bits of lime, which are uh, ubiquitous with Physarales, and I'm leaning Didymium as the genus here. Um, but maybe we can move on to the micro.
Yeah, I had one that looked like that, and I think Virginia, that's what she suggested mine was.
Oh, so the this is inverted, and there are species of Didymium that have a pseudothallus, and I think when I pulled this thing up, I pulled that pseudothallus up with it. So it kind of looks like roots on a tree. I'm pretty sure that's part of the Myxomycete.
Still waiting for it to load.
Those fingernails look dangerous.
Yeah, I stopped biting my nails a couple years ago and keep trying to grow them out to make them look dangerous.
You're doing a good job.
But they're the spores. They look like Myxomycete spores. We can move on. If you're not looking at them at 1,000, there's not much to
Oh, there you
be here.
go.
But here we go. Here are the crystals. And I didn't do a great job. I actually only did the micro on this earlier today. And right before the session started, I was like, oh my God, I didn't take a photo of any of the crystals. But they're beautiful under the microscope.
That was the last one for that.
Yeah, I think those crystals and the lime component points to at least the Physarales. Maybe there's something else in there. I don't know these things all that well.
What are those crystals? What are they for?
I'm not sure. I have no idea. But they are, I mean, you read descriptions of things in Physarales, and the frequency and the shape of the crystals is part of the description. So it's part of this Myxomycete. It's not just something that was in there, you know.
In general, like lime, it is sort of sanitizing, right?
Yeah, I think so.
Oh, is it
I'm
the
not
other
sure.
qualities? I don't know.
I took a class at the Eagle Hill, and there were similar questions asked to the person, you know, teaching the class, and he didn't have a good answer. So I'm not going
Okay.
to speculate. And then another thing on a sweet gum seed ball, and I
Oh
found a
my
mushroom.
God.
Yeah, I found a mushroom
Oh, that's
similar to
good.
this a couple years ago, growing and unquestionably growing from the seed ball. Um, and I called it not Tubifera, Tubaria, conspersa. And this, you know, looks like the thing that I found, and the research that I did at the time. This matches that. If you cycle through the photos, you'll see the yeah. You're gonna want to zoom. You can see that stretching like velar material, and then the kind of remnant of a ring zone pretty high up towards the gills.
Mm.
And then that kind of orangish color, the gills, that fits Tubaria. And then I took this photo just to, if you can go to the base, you can see that hymen or the basal mycelium. I mean, yeah, maybe this is a case of it crawled up on this thing, and that was the best place to fruit, because it is not like host specific to just these seed balls. I've actually in the past, when I think I've found the same thing, you know, you find it, yeah, it's on a seed ball, but it's also on some twigs and some leaves, and so it's just a generalist on hardwood material, whatever it is. Maybe it is specific to, what is it, Stylifera? Is that the genus for sweet gum? Um, but yeah, this one was clearly intended to be here on this thing.
Amazing.
Yeah, it's a cute little thing too.
For sure.
But yeah, I shared an actual mushroom. Anyone have an opinion on this? I mean, it. I think it's Tubiferous conspersa, but you know, I've only felt good about using that name one or two times in the past ever. So
Yeah, I have no idea.
I guess we're not getting anybody on iNaturalist helping us here, you know, and making suggestions, but I guess this is kind of new observation.
yeah, I think I probably uploaded it on Sunday. So, but I doubt anyone will.
I don't know what more they want from you. I mean, you got the micro for them on all these things and still no help. Right.
Would one be good for sequencing?
Yeah, I mean, I could just send it to Mycota blind, but I never hear. It's like a black hole sometimes. All right, and then here's another thing I'm pretty unfamiliar with the genus Simocybe. And I didn't include a spore shot here, but it's a brown spored thing. Growing on most likely maple. There were a few other smooth barked options here, but yeah, it had brown spores. The thing that I was looking at Simocybe for was the spore ornamentation, um, which can be kidney shaped or bean shaped, and that matched up. But yeah, I don't know. I don't know what this thing is.
Never even heard of Simocybe.
I've heard of it, but that's about it.
Yeah, and I've talked about it here before. It's one of those genera that whenever I don't know something, I'm like, let's see what the AI says. And it always wants to call something Simocybe. And I'm like, it's definitely not that. And this was the first time I was like, oh, I think it might be a Simocybe. And then looked on Myco go back and yeah, a lot of things match up.
Are those gills as thick as they are? I mean, the blades themselves look pretty thick. Or is that just a photography trick?
Oh, they're the biggest that are connected all the way to the site, and they're kind of folded over. So,
Mm.
they're not actually that thick, but they are kind of folded over. Like if you look at the forked one, you can tell it's kind of curved over. But
This one, just the fourth one. Yeah, okay.
Mm.
It's probably something we've all like walked by many times.
Mm. It's got a little bean to it.
The circle inside this spore, they don't seem to be consistent.
Mm. Yeah, they all seem to have the one, even the out of focus one to the far upright, you could see the gutule in there. Yeah.
I think Brandon is on mute.
yeah, what was
So do
your
these
question?
spores have
What was your comment, Sue?
the circles inside? Some of them look like they have two. Some don't have none. Some have none.
Okay,
Oh,
and What does that mean?
I don't know. Normally I see the spores are more consistent for inside, but I guess we're just going by the shape, not the content.
I think like
Right,
those
because
first,
that, like I said, that's what was in focus. I was focusing on the shape. But
yeah, when you look,
if you have information on the amount of gutules these are supposed to have, please share.
I have no idea. I'm not familiar with this genus at all. Though it is listed on Mushroom Expert. Uh, there is such a thing that it's been around for a while.
it used to be in something else. Do you know, Brandon?
He's
Oh,
back. He's
oh,
back on mute. Okay. Yeah. Yeah, the mushroom expert doesn't really say anything about the insides of the kidney-shaped spores. That's it on this last one.
Could you show the mushroom for that? I was kind of dozing, kind of tired.
A different one than the one I see. We'll go back to
For the bean shaped spores.
It's that
the
one.
spores.
Oh, it's this one. Okay.
Yeah, it's that mushroom.
Okay. Maybe Simocybe?
Yes.
What's so funny? I was falling asleep. I don't know what the
Yeah, it must have
discussion.
been in your subconscious. You heard that. That's where you're getting it. Right. But yes, that's what the
Or
proposal is.
or maybe he knows
Uh,
Simocybe.
they have bean shaped spores. They grow on wood singly. They're small. You know, it sort of fits.
Okay. Anything else, Brandon, that you'd like to show or comment on?
Nope, that's everything I sent you, Sue.
Okay. Thank you, Brandon. Um, Dave, would you like, Hunter, do you want to go or you want Dave to go next?
I can go. I'll go.
Okay.
It should be pretty quick. I'm gonna share my screen.
Okay, great.
Yeah, I had a good week. I found some black trumpets and some other things for the first time, and I ate Lactarius bulimus, which was real good. It was had a nice nutty flavor, and I really liked it. But photo wise, I didn't really get much. And this I've shown before. Uh, and I think it's a complex, but and I think I have the ID right. Lactarius subplinthogalus. It's the gills are really distant, and it exudes a white milk, and they're really and they grow with oak, and they're quite common in my area, and also in the south. They're very abundant, but they're supposed to stain pink, the milk after it dries, and I haven't really seen that. I did do some microscopy, and the spores are super cool. um this was fun to see. I wish the photo was a little clearer, but you get the idea. And I guess Lactarius in general, I think it's pretty entertaining under the microscope, from what I understand. Um, but that's really all I got that was worth sharing, photo wise of the
Are those spores mounted in melters?
no, just flaxing,
Huh.
like a real
Oh,
light wash.
oh okay. Because that usually you can only see the warts really well in melters, but I guess you've got
Oh,
some other trick.
oh, okay. Well, they did turn out well. Yeah, yeah. No, I didn't, cause that's the thing. So, again, this is a process for me, just figuring out what to do when, and that's something I don't know. And of course, I need to just look up every mushroom before I even start. But just all the steps, doing all the steps doesn't always happen. But right, knowing which chemical to do, because I kind of have one chance generally. I mean, I I should do maybe a few more slides with prints on them, but I often just have sort of one chance. I put the chemical on what I have material wise to look at, and it's just kind of like a guess, like kind of like, well, I'll use flaxine, I'll use water, or I've been using a little more Congo red, but I really don't know what to use when, except, like with amanitas to see if something's amyloid. So,
If you have melters, then all of the Russulaceae, including Bondarzuia burkellii, have spores with amyloid warts. So just the warts darken in the melt.
hmm
Then you can get an idea of any patterning as well, any
okay.
ridges that might be scattered or form a partial reticulum. So, but that's a good way to use up your melters really quickly because you know Russulaceae are very common.
Yeah. Okay. Well,
Those warts look like they're probably close to 2 microns long.
yeah, I think so. Yeah, they're really prominent, and it was a lot of fun. But um and this is a toxic Lactarius, I believe, as well. But yeah, I'm not. I'm pretty sure that's what it is, but I don't think
Usually,
anyone's.
as you said, usually you should get that salmon pink stain from the milk on the gills.
Yeah, I really. I mean, yeah, I don't. I didn't really. So
I think there's another species that has widely spaced gills like this. I don't have my book handy.
yeah, I
Um,
do not have a Lactarius resource really.
And
Now
it,
that I think about it, I do have those like micro cards for Lactarius, which are okay. Um, I don't know if they'd be in there.
if you also want to have, like you said, more than one chance, sometimes like if you position the slide so you're getting spore print across a large chunk of it, you could just drop multiple slip covers and then
Yeah,
just try different stuff under all of them.
yeah, I know. I should allow more, I should allow myself more options. But I mean, yeah, and yeah, I'm working it out. I mean, I'm just happy to be doing it at all, at all. Um, but yes, thank you for the suggestion. They slowly start to be practiced, so slowly start to be put into practice. So keep giving them to me for sure. So yeah, sounds good. So I got that, and then And I mean, everyone knows this mushroom, but again, it's just like practicing my process here. Um, so this is trying to open it. It's not opening. Oh yeah. Okay. So Exsudoporus, frostii, and I mean, we know that what it is, but again, I'm just going through the process to get it down. So it stained blue on the pore surface, and the bluing in this picture, it was rather faint. It actually blued more when I cut it at home later, which was interesting. I tasted it and I was really surprised at how I, it had a lot more. It was unusual. It was a lot different than anything else any of the other boletes I've tasted.
Was it
In
kind of lemony?
yeah, it was strong, and yeah, a little bit citrusy, but not necessarily like a good citrusy, but yeah, yeah, it was. Um, that's about as close as I could, say. But yeah, it was an interesting flavor. And I put KOH on the cap, and it turned this orange, which it is supposed to do. And I put ammonia on, and it really didn't do anything, which it is supposed to do generally, or it could have a slight reaction, but it didn't, and that's normal. And I just got some spores, um, which they're kind of, I don't know. They are. This is what I got. Um,
Fusoid.
yeah, yeah, right. So a little bit different from some of the other boletes I've seen where they're not as tapered, right? At the ends, they're more
Yeah, sometimes
uniform.
they're only tapered on one end.
Mm, this one's like that kind of.
Yeah, I wonder if it's just because of the way it's positioned, or maybe the other one's positioned in an odd way.
Yeah, and I unfortunately just have those two for some reason. Um, so yeah, so that those are the only observations. I did have um two photos. I found this hygrocybe. I have not much else to show except I tried to go through the key on Mushroom Expert, and I really got nowhere. But it was pretty. But I mean, this is the only photo I have, so it doesn't really give us enough information. And then this was really cool, and I just left it. But in retrospect, I wish I spent more time with it because I think I've found this before, and um, I can't remember what it was, where it grew in sort of like a cluster, and I mean, almost appeared to be bonded at the base, which may or may not be the case because I just left it there. But is it a trichoglossum maybe, or I don't know, or probably not. I don't
Well,
know.
this would be something useful. Your microscope would be useful on
Right.
these because then you can see if so I think Geoglossum, Trichoglossum, I think one of those two genera has the hairs on them, the setae. Um,
Yeah.
and so you can see that more easily with your microscope, but you can see those sometimes with your naked eye, I believe, because they're big. And then the spores. A lot of times, if you count the septa on the spores, that the number is always the same or close to being all the same, always the same. And sometimes you can get a species identification from your microscope.
Yeah, okay. Well, I will definitely plan to do that next time. Um, do you know of any that all grow out of one, like many growing out of like sort of in a cespitous fashion? Or,
I I I don't think I've seen this
okay,
with these kinds.
It's not
I
a,
okay.
don't
it's
know.
not
Maybe I
it's
just
not
forget, but
it's not uncommon.
not
It,
uncommon. Okay.
it it might not be a diagnostic thing. Um, I'm not sure that any of them are reported as always being cespitous, but I think a lot of them can be that way.
I
Um,
know I have found one at one point, and I shared it with you guys. I think like a year or two ago, and I thought that it was a defining characteristic, but I think I'm mistaken there. Um,
yeah, even in this one, which I do think I agree with you, Trichoglossum. I do think you'll probably find some hairs on the cap of these. They look the part. Um, but even maybe the three you can call cespitous, but that fourth one in the back, see, it's even separated a little bit, so
yeah,
they're probably not connected
it seems
com- com-
separate. Yeah.
completely. They're just kind of growing up against each other, perhaps.
Mm-hmm.
But
Okay. And then they were talking when I was looking into it about some of them the head is a
let
more separate structure.
y- yeah. And also, see, this is a great example of why these are all kind of so confusing. So if you if we assume that all four of these fruiting bodies are the same species, we have four different shapes of the head, and then we're going to have four different connections of the infertile surface to the fertile surface, that the head, quote unquote. Um, so that's yeah. These need to be looked at under the microscope, in my opinion.
Okay. Thanks so much. That's all I have.
Thank you, Hunter. Shihong, are you ready to go? You sent in the first email of the day.
Okay. Can you hear?
Yes.
Hmm. So, okay, last weekend I hiked at the Split Rock Reservoir. It was Sunday, right after our foray. On the trail, I came upon quite a bit of bolete. Uh, I don't know if Igor is here, but this one I'm pretty sure it's the Boletus separans.
Separans.
Yeah,
SEP. Separans.
separans. Yeah.
Yeah.
Nice.
If you go to next photo, I try to look at the stipe and cap junction. It does have that nut look things. And
Okay,
it was
everybody.
it was very fresh. No bugs at all.
That's a good find. Did you eat it?
Not yet.
They're best when they're sliced and dehydrated and
Oh,
used later.
so it should be dehydrated first. Okay.
Yes, I would slice them and dehydrate. Make sure you dehydrate. They're really well dehydrated. Like potato chips. Like you can, um, like break them apart, and you can store them in a jar, airtight jar for years.
Okay. So the next one, the color is different, but the
Is
This
Igor still
observation.
there? Igor, are you there?
Oh yeah, I'm here.
Yeah, is
Yeah.
that the real separans, or do you think that's that southern thing? Or
No, I think this is the real separans.
okay, mm-hmm.
It tends to be paler and not as
Taylor,
reticulated.
okay, yeah, because I thought it looked a little bit dark, but yeah, okay, that's why I'm asking.
Yeah, there are 3 species in the group, and we
great.
have 2. We have 2 in New Jersey at least. The third one tends to be more southern. The second species is Pseudoseparans, which is legitimate.
Oh,
So that one
name.
is also southern. Yeah, that one is also southern. Described from North Carolina by Smith and Thiers, or maybe just Smith and
Because you
somebody
know
else.
what, that species is actually documented with a picture. I don't know if it's correct, but in that old Phillips field guide from like the late 1970s or early 80s or whatever. Pseudoseparans.
Yeah, Smith and Grand. Those are the authors' names. Yeah, there's a black and white picture of Pseudoseparans in the paper that Smith published, but as black and white. But it looks quite dark there and very reticulated. And until we started sequencing these things, we all assumed they were all the same thing, which wasn't correct. But yeah, I believe this is the bona fide Boletus separans peck. And I'm going to stop there.
Okay, so the next one looks different color-wise, and also, but if you look close by the junction of the stipe and the cap, it also have that reticular. I don't know if the photo here show. AI gave me a different name. I just don't know.
AI can go and take a hike. This is
Yeah.
this is not Lexanum holopus. This is another porcini. You can see that from the reticulation, which is
Yeah.
white, and also the chylocystidia layer covering the pores. It's not separans. We have a number of choices like Atkinsonii, like Meripilus, which I don't think it is. Could be Nobilis.
Nobilis.
Yeah, Nobilis. Most, yeah. And there's also Leptocephalus, which is usually very, very dark and also reticulated. So yeah, I can tell you it's just probably an oak-associated porcini, Boletus, section Boletus, and that's as far as I would go.
That doesn't have a little bit of hypomyces on it, does it? On the lower part of the cap there?
I think this is just a spore print. Maybe
Oh,
not.
maybe.
That looks like hypomyces starting.
Nah, kind of I would say this is probably Atkinsonii, in my opinion. Fairly
Say that
pale.
again.
Yeah, it's not separans at least.
No, it's not separans. It's probably Boletus Atkinsonii, named after Atkinson, George Atkinson.
Okay.
Yeah, it's definitely not holopus. Is that what INAT
Yeah,
gave?
that's the top choice from AI. So that's
Great.
probably not. Right?
This is
Yeah.
great.
You're going to go correct that, Shihan, right?
Yeah, I will.
Okay.
Can you type in the species name?
Yeah, AI stands corrected.
Yeah, even if you just call it Boletus, if you, Atkinsonii sounds. I don't find that around here, but that seems to make sense to me. That's one of those grayish brown ones, right?
It's with a grain of salt. I mean, Atkinsonia tends to be fairly pale and has certain color of a cap that I cannot really describe.
A little bit orange, usually.
Yeah, what's the one? What's the one that has the yellowish band on the top of the stipe? Is that?
That's still separans. Used to be called Gertrudiae.
No. Oh, Gertrudiae. Okay.
Yeah, and that's synonymous with separans.
Oh, okay.
Is there a Chippewanensia or something?
No,
Chipuoensis.
that's edulous. That's edulous.
Oh, okay.
Yeah. Chipuoensis is another fancy name for edulous, which is a phantom species. Hmm.
So this one, the color look on top, the color looks similar, and the pores are a little bigger, and stipe smooth, and it's staining a little bit brown. Where my finger touches, a little bit brownish, Bruce. And this is a younger species, so I think it's Tylopilus.
Badiceps.
Yeah, that's the one that's supposed to have the beveled cap margin, isn't it?
Yeah,
Badiceps?
not always. Usually, like
Not
when
always,
the caps
okay.
get big, it sort of smooths out.
Oh, I see. That's what INET called it. Beveled. Oh, okay.
Yeah, I mean that's probably a good idea. I don't know how many species we have there. Really, we have to do a bunch of sequencing, but from what I remember, because I've sequenced a couple of these, they all came back to be the same species, and I called it Badiceps.
What about Ferruginous?
Ferruginous is more brown.
More brown. So this is more purple, I guess.
Yeah, this is
right?
more purplish. Yeah, and then the purplishness disappears. It gets brown, becomes brown too.
All right.
Sometimes you can see both colors present, transitioning from one to another in the same cap. Yeah, it's an excellent edible. Probably one of the best true Tylopilus.
Okay.
Yeah, but if you're going to eat Tylopilus, you probably want to nibble and taste just to make sure you don't have a bitter one.
That's correct. Right. Let me see what's the underside of this one. I have so many types, so I was getting confused myself. Okay, so this one, the pore is even bigger, and the stipe is smooth. So definitely not the Laxinium. So AI suggested a few things. So I picked this one, but I have no idea what should be named at all.
I'm not confident.
Hemloxanum sublabripes is fairly rare in New Jersey. It's mostly a northern species.
Yeah, that doesn't look like it to me.
Could this be Xerocomus tenax? Looks like it has a wide reticulum.
Yeah.
We have to see. It's just ridging, and we have to zoom in. I want to see the texture of the stipe a little bit better. The pores are definitely xerocomoids, so we're dealing with something in xerocomoidae for sure. Hemloxanum sublabripes doesn't have a smooth stipe, as the name suggests. It is somewhat Laxinum-like, and it does have scabers. There are like these scruffy points that are sort of densely covering the stipe. They are not like true scabers, but kind of similar, but they're much smaller and they're pale. This one doesn't have that character. Most likely it's some kind of a Xerocomus species, whether it's Tanax or something else. Tanax usually is pretty crudely reticulate. This is just ribbed. So I think it's just a Xerocomus. I bet you if you drop ammonia on the cap, it's going to flash green. I disagree with AI that this is Laxinum sublabripes.
Yeah, I wouldn't call this Hemloxanum sublabripes. Like you said, the stock should be like punctate.
Yeah, I would agree. It's not too, because I found that, and
the pores don't look right either, as you said. Xerocomus.
You can just write down there why, because this type is not scabrous. Instead, it's ridged, ribbed, pseudoreticulate, whatever you want to call it. And Sublaripes doesn't have that. Sublaripes doesn't have reticulations. And the pores are very large and angular, and they're xerocomoid, which points more towards Xerocomus rather than Hemloxinum. That too.
Okay.
I would say that H. subglabropes does not have a ridged stalk.
No, it
The
doesn't.
one you're looking at here is not truly reticulate. It's sort of verging on being reticulate, but it's not quite.
It's just, I would call longitudinally ribbed, prominent.
Yeah.
I don't see any netting, not a wide mesh or a small mesh. It's just ribbed, which is very typical of Xerocomus occasionally. I mean,
yeah, that sounds pretty sensible now.
10x is really coarsely reticulate, so I don't think this is 10x.
No, it looked at first like it might be verging on a reticulum, but you're right. It's just some ribs.
How big was this?
Medium size. I think half of my palm.
Really? That's pretty large for Xerocomus.
Yeah.
But 10x, or there's actually two 10x. There's 10x and there's 10x Indiana 01. Indiana 01 can get pretty large. We can get 4-inch caps for that one. But this is not 10x, I think. Yeah, and try to take photographs in natural light when you flash at them, or have incandescent light in your kitchen. That sort of distorts the colors.
That's right. I would take picture when I take them.
Yeah, you want to take a picture in situ,
In
or
situ,
like in a natural setting where you have a natural scattered light, no canopy, just like in the shade.
Okay. In the future, I will try that.
Yeah, because sometimes colors are also very important.
Yeah, in the group hike, I was just trying to keep up with everyone. We're still collecting mushroom.
Did you try to eat this one?
No, I haven't eaten any of the bolete yet. Because I also collect a lot of what do you call that? Hygrophilus, lactofluid
Hygrophilloides.
Yeah.
Right.
Widely spaced gills.
Yeah, we only saw one at Meadowwoods, one specimen.
So on this trail near Split Rock, there's so many mushrooms.
Too far out for me.
Too far.
Wouldn't this be like too mature to eat, or like I wouldn't eat this personally?
It's a little bit beat up, but it's not like post mature. I think it just was a little bit mangled in the basket.
Well, I would strip are you able to strip the tubes off of this, or are they really
the tubes are the most delicious part of the mushroom.
Oh, okay. Yeah, on a lot of the boletes, I don't like the way the mature tubes cook up. Well, like on porcini, when they're really mature, I usually get rid of the tubes.
Okay, I think that's it for you, Shahan?
Yeah, that's it. Thanks.
Okay, thank you. Does anybody want to go before we go on to Dave's email?
Yeah, I'll be right back. I just need a break.
Okay.
I'll be right back.
Igor, do you want to show something?
I could show something. I don't think I have much. I didn't take many pictures at Meadow, which had so many mushrooms. It was just no time to take pictures really. But there's one at least observation I'd like to share with you, and this is Shihong's. She found this interesting bolete at Meadowwoods that I happened to take pictures of. So let me load up MO, and I can share that with you. Okay, here we go. Right. Unfortunately, lighting at that pavilion is not the best. You either get too much sun or too much shade. And but can you see these pictures or this picture?
Yes.
Yeah. I'm just gonna flip through them.
What is the name of this one? I can't see the name, so.
Yeah. So the genus Butyriboletus in eastern USA is a small one. We just have a handful of species. And this is one of them. This is a fairly recently described taxon by Ernst Both. It's called Butyriboletus roseopurpureus. And in New Jersey, this is the only species that you find, and Meadowwoods is the only place where you find Butyriboletus in New Jersey, to my knowledge. I have not seen any Butyriboletus outside in New Jersey outside of that place. Butyriboletus is always reticulate, at least to some degree. It has a yellow pore surface that stains blue. And in the case of roseopurpureus, it's a reddy blue stainer. And as the name suggests, it's rose purple, and that refers to the color of the cap. So when you see that kind of a cap, and then you carefully look at the stipe and see this fine mesh reticulation that's fairly prominent, sometimes it's more subtle than that, you can rest assured that this is Butyriboletus roseopurpureus. There's nothing that looks like it. Not in that genus, not anywhere.
Yeah, the color, um, the stipe and the cap are of Brunius are different, a little bit different color.
Right. Yeah, and
Do you
Brunius
get
found.
Billier in southern Jersey, like maybe in the Pine Barrens?
It's a coastal species. There was a collection by Sigrid from Staten Island, New York, and we
Okay,
haven't found this. And
yeah, Luke found one also, but it might have been in Pennsylvania.
yeah, you found it in Pennsylvania, and we were supposed to go to that place this summer. Somebody's trying to organize a foray in PA with Luke coming in, and they kind of invited me as well. And I said, sure, I'll come along. But yeah, so Butyriboletus biliae is a coastal plain species mostly, but you can apparently find it more inland, but it's pretty rare. It's also in Mississippi, and it goes as far north as Cape Cod, from which it was described. But Butyriboletus roseopurpureus is more widespread. I think it goes as far north as South Canada, certainly upstate New York.
I've never found it around here.
They also have it down south, like I said, in Mississippi and maybe even Florida. Yeah, it's sort of not a very common species.
Igor, if you click on the link I just put in the chat, that's Shihong's in situ pictures, which are fresher.
Would you like me to share that?
Yeah, you just click on it just so we're in context.
Yeah, I'm gonna then stop sharing this and share INET. Yeah, so this species is hardwood associated, whereas Butyriboletus bruneus is a hemlock associated species. As you can see, it stains readily on the stipe, and from the previous pictures you saw that it also stains on the pores. And you can also see, as it sometimes happens with yellow pored boletes, that when they attain a certain age, when they're older or when they are desiccated, the pore surface also is tinged with red, and you can see here it's reddish in certain places. So don't be fooled by that. It's a character that's not constant and only happens, due to the age or the elements, weather elements. Right.
Thank you, Igor. You got others to show?
Maybe one more.
Okay.
Yeah, on my way home, I stopped at a undisclosed location in Somerset County, and I found this Amanita, which I haven't seen there before. I thought this was maybe Anusta, but this was way too big. This was like 4 inches tall. I took 2 pictures, and you can see that it has a very robust partial veil that's sort of coming apart, and you can see the appendiculations on the edge of the cap as it rips apart, and there are these mighty warts both on the cap and at the base of the stipe. I did not excavate this. I just snapped off a piece of the cap and dried it, but I left the mushroom essentially there untouched.
What about Scenario penosa?
Yeah, I thought about that, but I haven't investigated, the appearance of this. Yeah, there's Scenario penosa and there's a Scenario conia. I always confuse them.
Oh, okay.
Yeah. So, but at least I got a part of the mushroom that I can submit for sequencing, and interestingly, that place, which is usually loaded with the mushrooms when it's wet, was completely bereft of fungi. I only found 3 amanitas, no boletes, and this is just one of them. So compared to meadowwoods, this place apparently did not receive as much rain. So yeah, it was pretty large. It looks similar to Anusta, but Anusta is much smaller. I mean, this thing was 4 inches already, as I said, and it's hasn't even expanded its cap. So probably it's not Anusta. Whether this is Scenario conia or Scenario penosa, we'll find out maybe this year. And that's all I have to show for now. I have more, but I'm going to let other people present their stuff.
And Lila reported that she lost her internet, and we seem to have lost a lot of our participants also. But Dave is still here, so we'll continue on.
Wow, 8 people down from 16.
Yeah.
I wonder why.
Well, the internet went down. That's at least for Lila, so I assume others might be affected too. But okay, I will share.
The provider, right?
I don't know if it's the provider. It could be a power. Well, Lila did not report a power outage, only that she lost her internet, and I don't know who her provider is.
Who's yours?
Fios. I live Verizon.
Okay. Verizon. Okay. I have Comcast.
Okay. Yeah, it all depends where your node is. Okay. Share and go on to Dave. Dave, you're still here in spirit, right?
Yeah. I'm here. Yeah. Yeah, actually, I'm not dozing off right now, so I might have another 20 minutes or so left.
Okay.
Lots of mushrooms here. Lots and lots of mushrooms right now. The oak woods actually is where things are really happening. So Pseudosperma is an offshoot of Anaceae. It's a genus in the Anaceae. So you look at this and you say, well, that looks like an Anaceae to me. Looks like an Anaceae to me also. It's a fiber head, right? Look at the way the cap is. radially splitting into sort of fibers. So why is it that I would think that this is pseudosperma and not inocybe? It's you have to use a microscope, and it's the gills. You have to smash mount some gill. Why smash mount? I mean, if you're really serious, you try to do that Roman aqueduct thing that I can't get the hang of. But with the Inocybaceae, you get away with a smash mount, and I think I can explain why once we get to the micro pictures. So first we'll see some spores. The spores are, I guess, let me see. Look at the gill edges. You can see on the gill edges, they're kind of coated with those are Chylocystidea. So that's actually a clue, because pseudosperma has Chylocystidea, a lot of them, but no Pleurocystidea. So that's kind of a clue right there. But we'll see better in the microscope what I mean. So the spores are kind of just sort of large and ellipsoid. I don't know. I guess you would say these are almost elongate. Looks like the Q is probably close to 2, but a little bit smaller than 2, maybe 1.9. Maybe a few of them, the Q is over 2. So that's not enough to rule out inocybe, because there are inocybe species that have ellipsoid spores. So what is it then that your microscope? Well, here we see part of the gill edge. Now, how do I know this is the gill edge? Well, sort of backwards thinking. Since I just do a smash mount, I'm not really keeping track of my mount of where the gill edge is. But when I scope one of these Inocybaceae, and the only place I see cystidia are sort of a whole bunch of them grouped together, bunched together along one edge of the mount. And I can be fairly well assured that those are Chylocystidea on the gill edge. And also, we saw the close-up of the gills, and you can see the gill edges were what is sometimes referred to as crenate, meaning they're just kind of, it's not quite toothy, but grainy sort of. That's the Chylocystidea. So this is a bunch of Chylocystidea right here. They're a little bit buried in the gill. Maybe the smash mount actually partially buried them. But the other thing is that you don't see here that I did was I searched the mount of this smash mounted gill and did not find any Pleurocystidea. The Pleurocystidea are usually have much more interesting shapes. They're more isolated. They don't form what they sometimes call a palisade. So the Chylocystidea form a lot of at least some of the pseudosperma species form a palisade. So you might also wonder, how do I know this isn't inosperma or melocybe? I'm going on morphology to eliminate those two. And I think this is probably Pseudosperma rhomboides, I think is the species. That would be my guess. And there you can see maybe a little bit better look at the palisade of Chylocystidea. And they just look like balloons, sort of. They're not particularly interesting looking. Like when you get a true inocybe, which we'll see next, because that's the next observation after this one, is going to be a true inocybe. And the mushroom doesn't look all that much different. It's got this head, this cap that's a fiber head, right? It's splitting radially. And so it looks, fairly similar to the last thing we saw, right? Color's a little bit different. The stipe is a little thicker, a little bit more robust, maybe. But it's once you get to the microscopy, then we're going to see some striking differences. So there's your gill edges. Those gills look a lot like that pseudosperma, except the gill edges they're not crenate. Also, we have these little scales on the top of the stipe, also near the apex. That's kind of interesting. I'm not sure if that's if that trait is strictly an Inocybe trait, or if you have other genera in the Inocybaceae. I think maybe there are some non-Inocibes in Inocybaceae that have this sort of pruinose stipe apex. But it gets really interesting when you get to the microscope. That's where these guys are interesting, these Inocybaceae. So that should be coming up soon. Whoops. No, it's same. We just need to go through the photos because I know I have microscopy.
Yeah, I'm just trying to get to back to the original.
Oh yeah, you have to go back on your back to the last window thing. Okay, well you're good now.
Yeah.
So,
so we're going to go to.
It looks like the 6th photo there. Look at these spores. If you can zoom that, it'll zoom a little bit because it's cropped, so it's not going to zoom a whole lot, but it'll zoom a little bit if you use the zoom feature, which now it's the mic. Yeah, there we go. Look at these nodulose spores. Not all true Inocybe species have nodulose spores, but the converse is dependable. If the spores are nodulose, then you have a true Inocybe. The other genera that are found in eastern North America, Malacybe, Pseudosperma, Inosperma, none of those occur with nodulose spores. So these spores are quite irregular. They're these little like bumps all over them. Those are the nodes, right? So these are nodulose spores. So as soon as you see that, you know you have a true Inocybe. But you still might have a true Inocybe even if you have ellipsoid spores. So what then do you depend on? Well, then it's the hymeneal cystidia, meaning the cystidia that are found on the gills. So yeah, what you do is you go back to the last window now, and you'll be back. Yeah, now you're back in. Now you need to go
Well,
back.
I'm back to an image. I'm not back to the observation still.
Yeah, you need.
Now I have the back button enabled.
Yeah,
Before I did
that's
not
it.
have
Do
the
that.
back.
Then you go back to the observation. Right. So now we'll see these funky sort of cystidia that they have more interesting shapes than that palisade of balloons. Right. So this thing's shaped kind of like a bottle. It's got an ornamented apex, so it looks like it's got this little furry hat on. And the walls they should be thick walls. If that's not coming through really well in this photo. This one here, you can see the walls are a little thicker, and it's got the ornamented apex. And these are more or less all over the gill mount. You can usually find these pleural cystidia all over the gill mount. So they're not like restricted to one edge, like the chylocystidia would be. So a true Inocybe, don't ask me what the species is. There's only a handful of Inocybe species that I'm comfortable with. I try, claiming an identification to species. Now you see here toward the apex, you can see where the wall's a little bit thickened, right? Where there's like that gray part along the perimeter, like that's the thickened wall, and that does drop down a little bit, especially on the upper part of the photo. You can see so that's like a thicker wall. So the pleural cystidia generally have thicker walls than the chylocystidia, and they call those things metuloids, those thick-walled pleural cystidia. So true Inocybes have metuloids.
That was the Inocybe.
Yeah, that was the true Inocybe, and the first one was I'm pretty sure Pseudosperma. So this is a cool little thing. I found it in my yard years back. I don't think I've seen this for over 10 years. Calocybe carnea. It's a pretty little pink mushroom that grows in a little group on your lawn. I just haven't seen this species for a long time. I have seen it in my yard before, not in the exact same place, but on the lawn. And it's a pretty little mushroom. It's not particularly interesting, with the microscope. Or I don't know. I didn't look for cystidia, so maybe it does have interesting cystidia. But I knew what it was. I knew what this was just based on the morphology. But I did sample it also. I sampled it for sequencing. So those are pretty cool. They're just.
What is this one? I had to step away.
Calocybe or Calocybe carnea, I guess is the way to say it. Mm-hmm.
Neat.
Yeah, it's a neat mushroom. It's that's why I'm sharing it. It's just kind of a cool thing to find.
Very nice.
Yeah.
And we assume this is a saprobe on the lawn. It's not.
Yeah, these are saprobic. They use this. Yeah, these are saprobic. I'm pretty sure. The spores are not particularly interesting. They look kind of like a lot of small saprobe mushroom spores. I didn't do any other microscopy on this. I've been collecting a lot of mushrooms, and sometimes I just don't have time to do microscopy.
So I hope to see this one. This seems nice.
Yeah, these are nice to find. They stand out because of the color.
I'm wondering if it likes the grass or likes the weeds.
I think it likes the grass. I think it's probably fruiting from like old grass roots or something. So there's two in a row here, and I think they're the same thing. And it's just a really impressive, what you might say is a grisette, because it's Amanita section vaginate. But look at that umbo. That umbo is very hard, almost like a rock. Because this species, which I did not identify the species. I identified it to subsection. I think it's subsection. Penetratrices. So the mushrooms in subsection penetratrices of section vaginate originate from pretty deep under the surface of the ground, and they use this central umbo to push up through stuff that's in the way, like little rocks and things, maybe a root of a plant or something. These are really big. Now, the interesting thing here is these two little guys, and I probably should have taken one of them home. I'm assuming they're the same species as this big one. But I could have at least taken one of them home and looked at spores. Although a lot of times with section vaginate, the spores all look the same. They're globose, and they're like, 11, 12 microns max. Something that. I did get spores from this one, I believe. Or did I? Maybe not. No, it's the next one. There's another one. So I found also another one of these great big, and what does the ID say? Not down below, because I proposed something else also. Because I can do better than section vaginate. So down below the photo, there's, if you just like scroll down to where, yeah, there it is, subsection penetratrices. So what's the species? I don't know. It could be, there are 3 different species, I believe, that I have found over the years that I sent to Rotulas, and he analyzed and named them. And so there's penetratrix, penetrans, and pseudopenetrans. And I believe I found all 3 of those over the years, but I'm not sure how to tell them apart, just from appearance. But you can see here where this stem is pretty well rooted in the ground. And so you do have to be careful the way you dig these up, or you'll lose the basal features. So you have this membranous basal volva like you have on a lot of vaginate. It's just, these are really big. These guys are really big. The one picture, I think I put it there. There's the penny. You can see how big this thing is. So the next observation, you can just go right to it. I think it might be the same species, but it's a different color. The cap is a somewhat different color. That one, and that color was, that gray color that you saw in that last one was pretty true to form. And this one's really kind of a light brownish ochre yellow sort of color. Once again, you see the great big round umbo. It's a really big mushroom. It's rooted in the soil. Oak woods, the other one was in oak woods also. And see, I put a penny there. You see the penny? So you can see how big this thing is.
Yep.
It's pretty big. And this one I did get spores. Like I said, you don't learn a lot from the spores because similar to lots of species in section vaginata, the spores are just round. But there you can see there that, this is a very large mushroom. And I did have to dig it up to make sure I didn't lose the basal features.
I would tentatively ID that as penetrans.
Penetrans? Okay. Well, I sampled it, so maybe we'll find out.
You will find out.
And so there's spores coming up, but I just wanted to get a few photos here that just show how big the thing is. It's got the sort of the ornamentation on the stipe, the sort of almost like a chevron pattern. But you can see the spores here are more or less round. There's one or two of them are a little bit, maybe subglobose, maybe verging on broadly ellipsoid, but I would say more like subglobose. Typical for the section. So these are always fun to find because they're so big.
So nice.
And like all species in section vaginata, it has the cap margin that's striate. I'm not sure if the photos are really showing that well enough. You can sort of see it here.
Yeah, you can see it on the upper left for sure.
Yeah, they're actually pretty long. It looks like a 50% of the radius. I got one more thing here, I guess. And this was just absolutely striking to run into these. There was one other great big one too. But Amanita jacksonii, the two smallest ones, those were allowed to remain in situ, and I got two other ones and posed them. And this is what it looked like. I mean, this, I did fool around with editing a little bit to get the picture to be more clear and sharp, but this is what they looked like. This is the color. The one on the left did have more or less a yellow margin. That's true to form. They have this orange partial veil that forms a skirt-like ring. And the thing that really gives away this species, other than the really bright scarlet sort of cap color that's striate, definitely striate, like section vaginate, but section caesariae generally has a prominent partial veil that forms a ring. So, but striate cap margins and these really, really prominent basal vulvas that pretty much all of the universal veil is down there on the bottom. So it doesn't really leave much at all on the cap, if anything at all. The whole thing, the basal vulva, just ends up on the bottom of the stalk. And by the way, if you want to eat amanitas, you have to really make sure you know what you're doing. But this species is edible. I ate the two small ones. The two bigger ones I replanted into the soil, and they'll continue to expand. Amanitas do that. They'll continue to expand. They'll drop their spores. They'll be perfectly happy replanted right near where they were to begin with. But I took these two smaller ones, these more or less just beyond the button stage ones, and they were really good. It was a very, very tasty mushroom with really good texture. So that's
Is
Amanita
there any
jack.
reason to take the vulva?
Oh, I'm
Is
sorry.
there a
Go.
reason to take home the vulva? Is there a reason to take home the vulva at all? Do you eat the vulva?
No, I didn't eat the vulva because the vulva has a lot of dirt on it. So I took
Okay.
them home with the vulvas on them. But then after I looked them over to prepare them to put into a skillet, I got rid of the basal vulvas because there's a lot of dirt that just clings to the basal vulva, and I didn't want to fuss around with trying to brush it off. So I just tore the vulvas off.
In general, does the vulva have any flavor that's useful?
I don't know.
Okay.
The mushroom itself it's not a really it has a nice flavor. It's rather subtle. It's not a real strong flavored mushroom. Now they say when these get old, they develop a strong flavor that's somewhat unpleasant. But the two that I brought home, those two small ones, they were really good. And they were all pretty big too. These are good sized mushrooms. Somewhere I guess I'll have a picture here with a penny in there. I think I must have done that at some point.
Maybe the first one
Maybe
had a
not.
penny. There it is.
Oh, there we go. Those are the two
Small
small
ones.
ones. Those are the small ones. So, yeah, and you can see the stalk has the orange ornamentation on it. That's one feature that helps to separate Jacksonii from some of the other scarlet or red-capped Caesars that tend to occur a little bit more to the southeast or maybe the southwest. There's one called Arkansaniana, I think. It's from Arkansas. But those tend to have not as much ornamentation, or maybe a bare, even almost white stipe. But Jacksonii has this stipe with the orange flocose ornamentation on it. Yeah, so there's the penny. Yeah, so that's it's really something. So there were some guys. This was in a state park in Rickus Glen State Park, and there were some workers there who were, I guess, they were doing trail maintenance or something. And the one guy saw me, examining these, and he said, like, "Oh, I've been watching these for a couple of days. What is it? What is it?" He wanted to know what it was, so I told him. But these are impossible to not notice when they're out there.
It's funny that we don't get these in New Jersey.
I think they might like hemlock. The places where I do find them, there are hemlocks present.
Yeah, but these are found in the deep south, where there's no hemlock.
That's probably the other species.
No, I think it's still Jacksonii.
Jacksonii?
Yeah.
Oh, okay. All right.
It's both distributed to the north of us and to the south of us, and New Jersey doesn't have it. I mean, we have Baningiana, we have Spreada, and we have other sections in this area.
Yeah, we have Baningiana also, which I've eaten that one also, and it's pretty good. It's a little different color, but a lot of the features are more or less the same. The stipe is not ornamented, and I think the partial veil is not quite so pigmented. It might actually be almost white, but you have the big basal vulva. Not quite as big as on these guys, but it's a pretty prominent sake basal vulva on Baningiana also. Yeah, that one's an oak one. Baningiana likes oak.
I think that's the last one for you, Dave.
That's it for me. Yep, that's my 5. Thank you.
Okay. Well, does anybody else have anything they'd like to share? There's almost no one left because the internet outage from Verizon. So we'll call it a night. And we'll find great things this week too, because we still have more rain that has fallen over the weekend.
Yeah, you know what? I wanted to mention, Igor, you probably have noticed this also. There was quite a bit of rain in the Mont Alto area over the past like 5 days.
Yeah, I've seen some rain and more is in the way, so this is good news.
Yeah, yeah. And there's supposed to be a rainy week next week in Pennsylvania.
That's
So
what I'm
we'll see.
saying.
Yeah.
All right,
Yeah.
so the
so-
ninth event in Mount Alto will be great.
What's that?